Thank you for joining us today. My name is Aleksandra Babicheva from University of Minnesota. Today, ATS Assembly, Respiratory Structure and Function present live webinar that will feature 2024 Robert Crapo Memorial Innovative Research Methodologies Award winner. And we are fortunate to have Dr. James Crapo, the brother of the late Robert Crapo, who will give introductory remarks concerning his brother's impact on respiratory medicine. So, Dr. Crapo, welcome.
You ready? I will share my screen and just give you a couple of pictures of Bob to make this a fun introduction. It's my brother Bob, and it's a real honor to be asked to be here to say something about Bob. I was 2 years younger than Bob, and but I've been admiring and following him from a very early age. And I would just say this is a picture from the late 1940s. I was already following him at that time. And I'm just going to give you a couple more pictures. This is when in the 50s, when obviously I followed Bob into do scouting. And then from there, turns out that let's show one more picture first. This is a picture from the 80s. It's my brother Bob on the right and my mother. And on my far left is our younger brother, Mike. Mike didn't choose to go into medicine like Bob and I. He chose to go into law and politics. And he's actually a U.S. Senator right now. And next week, he'll be chair of the Senate Finance Committee. It's important because he actually has made a commitment to Bob and I to represent COPD, the caucus in Congress, and push for funding and support for building an understanding of chronic lung disease. So we'll see some of Bob's legacy go on, hopefully for the next few years. But Bob chose to go to the University of Rochester for medical school, and I followed him there 2 years later. So we spent wonderful early years of our training together, and that was in the middle of the Vietnam War era. So Bob chose to go into the Navy, and I thought he had told us his goal was to be in anesthesiology. So he went into the Navy and spent 2 years in the Pacific on a ship as a medical officer. And I chose to go to the NIH and start doing research in cell biology. And we really didn't cross paths again for about 4 or 5 years, both on different sides of the country. When we got back together, we discovered we were both in pulmonary medicine for very different reasons. Bob had loved intensive care medicine and decided the real way to do it was through pulmonary medicine. And so we switched and trained there at the University of Utah. And actually working with Alan Morris and several other people there, he built in the early 80s one of the most incredible intensive care units in the world. Now, I'll tell you, when we went to Rochester in the late 60s, there was not an intensive care unit at the University of Rochester. So intensive care was just developing. And when I got back together with Bob and found out what he was doing, I asked him, wow, that's so complicated. Why are you choosing that? And he said, intensive care medicine is really very easy. We just put lines in everything, and then we measure it, and then we normalize it. Now, it's somewhat simplified, but that epitomifies what Bob did with his life. Because what he found was that when you try to normalize it, there were no normal standards for anything. None of the things he was measuring had anybody develop standards to say what normal was. And his compassion in life was to really address that question. The first thing he did was to work with his group there to develop functions, what's called the Orange Book. I still use it. Maybe some of you have seen it. But it was the first really well-done set of pulmonary function standards that were published and then used. What was interesting is that the most major manufacturers of pulmonary function equipment quickly started using those standards and embedded into their software so that when you printed out your pulmonary function in any city in the world, you got Bob standards. And I'm on the East Coast now, thousands of miles away. I would go to meetings and community hospitals, and people would come up afterwards and want to talk to me and tell me that their pulmonary function equipment was named after me. And what they meant was, I found, that the manufacturers had put the word Crapo at the bottom of the pulmonary function test to tell people what their standards came from. So I had to tell people repetitively in my lifetime that was my brother. And we had this wonderful relationship over time. The um, the Bob went on to be the chair of the ATS Pulmonary Function Committee for the ATS to set standards. What he quickly discovered was not only was the standards necessary, but then compliance, quality of performance or proficiency was required, because if you didn't run your lab well, the standards wouldn't mean anything by the way you use them. And that became the dominant theme of his life and many of his work and his publications. In fact, when the ATS and the ERS merged merged a lot of their interest in the 1990s. Bob chaired the international committee to look at pulmonary function tests and proficiency standards worldwide. So he had a lot of impact, but his big impact was driven by this desire to have rigor in medicine and set standards that let you interpret what you're getting from patients. And he believed solidly in the use of numbers and that rigor that came with it. Now, I would say that Bob was also had a lot of other interests. He loved living in Utah. He loved mixing his work with this is a skiing that was in the air, the whole outside area. But in reality, I think that his big love over everything was fishing. And he tied flies and made fishing poles, and some of my best memories of that are from things he's left for me from that. But he lived a very full life. Unfortunately, about 10 years ago, he died with Alzheimer's disease. And it was a big loss for the community. But he left a legacy that started in the very beginning. In fact, I think the 70s, the 80s, and 90s were really a reformation of modern medicine as we started getting access to all kinds of data. And he led that in pulmonary medicine and the pulmonary function unit, or the intensive care units, to use rigorous data and rigorous techniques to make good patient decisions. So he really laid the foundation for what we'll talk about today. And it's wonderful that the ATS has dedicated this memorial lecture to that thing. And I think that he gives a legacy that you'll see amplified now 50 years later. So it's a real honor for me to represent Bob and just tell you a little bit about him and open this lecture now for Dr. Krishnan. Thank you very much. I'll stop sharing.
Thank you very much, Dr. James Crapo. So next, I would like to introduce Dr. Krishnan. So the number one, I would like to talk about the award itself. So the award is in honor of the achievement of the Dr. Robert Crapo, which was an international leader in pulmonary function diagnostics, like the Dr. James just mentioned. And basically, he focused on a novel approach for standardizing the lung function. So the following this track, basically this award gives to the best researcher in the respiratory structure function. That is the researcher focused on the new technology or the new methods to measure lung function or the structure of the lung. Among many awardees, we have Peter Noble, Jason Bates, David Kaminsky, Zoltan Hantos, and Charles Irving. And today I have a pleasure to introduce Dr. Raman Krishnan. So Dr. Raman Krishnan did his bachelor in mechanical engineer in India at National Institute of Technology. And then he came to US and did his master's and PhD in mechanical engineer at Columbia University. After his degree, he decided to go to Boston at Harvard School of Public Health, which he spent a few years doing his fellowship. And then he was awarded in the meantime of the prominent Parker B. Francis Fellowship in Pulmonary Sciences. After that, he was appointed faculty at the Harvard Medical School and in the Beth Israel Deaconess Medical Center at the Department of Emergency Medicine. And he's being worked as a faculty. Right now, Dr. Rama is an associate professor and it's being published over 80 peer-reviewed manuscripts. as well as on top of that, consulting for many different companies, and he's entrepreneur by himself. Thank you very much, and I will pass now to our guest of honor.
Thank you, David, for the introduction. Dr. Crapo, that was an excellent introduction to everything I want to say. And I hope that I can convince you by the end of this talk that my theme, as well as pulmonary function measurements, I carry forward this torch. I carry forward the theme, but not in the whole lung setting. But you will see measurements at the level of cultured cells, at the level of tissue slices, and so on. But a common theme across almost every slide is functional outcomes, functional measurements in disease settings. So with that, let me start by disclosing my commercial interests. I've co-founded several companies, and I'm sorry. And David, you mentioned this. I consult for others, but none of this is going to impact the science that I'm about to present to you today. So for the uninitiated, I like to start my talk with an exercise. After all, it's pulmonary function. It's a lung function. So what I would like to do, all of us in the audience, and I would like for you to participate. So when I count up to 3, I want all of us together to take a single deep breath. Okay, so 1, 2, 3, please. Feels good, right? Well, it turns out you do that every 6 minutes or so. And every time you do that, your airways that can sometimes constrict will immediately dilate. Of all natural strategies that your body uses for bronchodilation, that deep breath that you took is amongst the most effective. However, it fails in asthma. Now, this is not by any means anything I have discovered. It's been pursued by leading experts in the field of airway smooth muscle biology, physiology, lung mechanics, lung function testing. And these group of investigators, accomplished investigators, have discovered many different mechanisms: airway smooth muscle mass, airway smooth muscle tone, contraction velocity, inflammation, parenchymal tethering. But a common mechanism has remained elusive. To elucidate such a mechanism, I turned to human lung slices. This is about 200 microns thick. It's a 250 microns thick slice of a human lung placed in a dish. The 40-odd cells that are present in our lungs are present in here. They're living and they're interacting. And what I'm showing you here, that's the airway. You have airway. There you have the epithelium on the inside, muscle on the outside. And I'm going to run an experiment. And I want you to keep your eyes on that airway. So first I add methacholine. And in response, you can see bronchoconstriction. And then I superimpose stretching that mimics breathing. Oops, sorry. So what you should have observed there is, so in response to methacholine, you see the airway narrowing, but with breathing, it started dilating. In fact, if I waited a bit longer, this would potentially return back to our pre-treatment value. Don't take my word for it. We've done this many lungs, many slices, many airways, and I'm showing you here average data. This is percentage reversal of constriction on the y-axis and the amount of stretch on the x-axis. Differences between these curves is not important for the talk today. What's important is the more I stretch, the more reversal of constriction you get. So that is to say, this is like I can reverse my maximum bronchoconstriction state. Now, all those studies were performed in non-asthmatic, healthy human lung slices. We have data accumulating now in asthmatic slices. Here's one such experiment. Same thing, methacholine leading to constriction and then stretching. That airway stays constricted. Stretching does not dilate this asthmatic airway. So now you're probably wondering, what's going on here? Let's turn our attention to that airway smooth muscle around the airways, and especially its contraction. For this representative human airway, that's the muscle around the airway, I'm showing you here maps of those contractile forces. And these are forces that are exerted in response to methacholine. what we will do now is we will take that slice and we will impose this protocol. So we will do a big stretch, unstretch, which resembles a breath, and then breathing, and then no breathing. Well, no. So inspiration, expiration, and then I'm going to play a movie where the first couple of frames are going to be in this region A, which is prior to stretch, and the remaining frames are after the stretch, unstretch maneuver. So keep your eyes on that movie. So here, pre-stretch, immediately after stretch, and with time after stretch. We can be more quantitative about this. From every such map, I can get a single number for contraction. And I'm showing you how that contraction changes over time. These points here is in that region A. So prior to stretch, well, it stays contracted with methacholine, nothing happens. But then this point, and all these subsequent points are in this region immediately after stretch. So what you can see is soon after that stretch, unstretch, forces dramatically reduced by as much as 80% for this particular slice and experiment. But in time, things recover back to that pre-stretch baseline value. From such experiments and many others that we've performed in cultured airway smooth muscle cells, muscle strips, intact airways, and even human studies, we've now come to learn that in response to stretch, the airway smooth muscle cell fluidizes. And by fluidization, I'm referring to a physical phenomenon that is characterized by reduction of contractile force, cytoskeletal softening, enhancement of molecular rate processes within the muscle cell, and these collectively promote dilation of the constricted airway. And we've hypothesized that in asthma, it is the failure of the airway smooth muscle cell to fluidize, which is the proximate cause of bronchoconstriction. At a molecular scale, we've also learned that this fluidization response is mediated by F-actin dynamics. Let's go back to my favorite stretch protocol. This time we ran it in a dish of cultured cells. So we took cells and fixed a dish of cells immediately after that stretch, unstretch maneuver. And I'm showing you here cells outside in the same dish, outside the stretch region and stained for F-actin. So that's the F-actin staining outside the region. Compare that to staining inside the stretch region. It's day and night. Over many experiments, many dishes, what we've learned is immediately after that stretch. So at this point here, you can see the F-actin reduces dramatically. We then took another dish of cells and this time fixed it here. All of that actin recovers. So over time, all this F-actin reassembles and recovers back to the pre-stretch value. We also confirmed these data with Western blots. Compared to no stretch, and here I'm showing you G and F-actin, compared to no stretch, we notice immediately after stretch, you can see the F-actin band is almost invisible. It's very hard to measure. But 2.5, 5 minutes after stretch, everything recovers back. We went deeper. We first focused on the actin depolymerizing protein cofilin, our favorite stretch protocol, and we measured contractile force dynamics. Compared to control cells, shown here, we noticed that cofilin knockdown cells relaxed significantly less. So compare here to there. But in time, everybody recovered the same baseline value, which is to say that this acute force reduction after that stretch maneuver is mediated by cofilin. We then focused on the actin repolymerizing protein zixin. We now observe that compared to wild-type cells, shown here in blue, the zixin knockout cells relaxed to the same extent, but they recovered substantially less, which is to say that the force enhancement after stretch is mediated by zixin. Now, zixin, so cofilin is here, zixin. Zixin, cofilin are but two proteins in a huge, large number of proteins that are associated with the focal adhesion of the airway smooth muscle. So I've just tip of the iceberg here. There's many other proteins that could be therapeutically important and that remain to be studied. However, beyond the focal adhesion, there is now a number of studies that are focused on intra and intercellular proteins in the airway smooth muscle that can mediate effectin dynamics. So these include transgelin, ROCK, ROA, integrins. I'm going to focus on this one, N-cadherin antagonism. The conventional physical picture of how N-cadherin functions in the airway smooth muscle is as follows. The N-cadherin will bind to beta-catenin, cluster actin filaments, and this then will permit forces to transmit from cell 1 to cell 2. That is to say, N-cadherin transmits intercellular forces. This is the conventional picture. From direct molecular measurements, what we have recently learned is that N-cadherin can also bind to delta-catenin and thereby directly regulate F-actin polymerization and ultimately airway smooth muscle contraction. So this was not previously studied in any length, in any detail. And what's interesting is this particular pathway of force generation through N-cadherin is independent of canonical excitation-contraction signaling pathways. To examine its therapeutic implications, we considered ADH1, which is an N-cadherin antagonist, a small molecule compound that has already been validated as safe to use in humans. It's successfully passed phase 1 clinical trials. We started with cultured airway smooth muscle cells, and we compared vehicle-treated with ADH1-treated airway smooth muscle. What we noticed that ADH1-treated cells had significantly smaller muscle contractile forces. So you can see, compared to these large red hot spots that represent regions of high force, in the ADH1-treated cells, you see mostly blues and greens with much smaller forces. And this force reduction also stood in the face of additional treatment with the contractile agonist histamine. So compare this is pre and post histamine. You can see enhancement of force in vehicle-treated cells. Compared to ADH1 treatment, you look at, there really is no force enhancement. In mouse lung slices, we observed that pre-treatment with ADH1 substantially reduces bronchoconstriction to methacholine. In human lung slices, we noticed that ADH1 treatment reduces contraction in response to 10 micromolar methacholine. So red is donor 1. Here is where we treated ADH1, and you see this nice reduction in response to treatment. But what's also interesting is we did some additional protocols here where we desensitized these airways to formoterol, which is the current clinical treatment. And you can see first after methacholine, you see the contraction, but then when we treat it with formoterol, nothing happens in either donor. So these are desensitized. However, ADH1 continues to function. So it can work even in the face of beta agonist desensitization. We then turned to mouse experiments. We measured airway resistance in mouse lungs in response to increasing methacholine dose. First, let's compare to vehicle, PBS-treated mice. Those mice that were sensitized and then challenged with the fungal allergen Aspergillus had enhanced airway hyperresponsiveness. So compare the green with the blue. You can see this big difference. Treatment with ADH1, acute treatment just for 24 hours, can substantially reduce this airway hyperresponsiveness in mice. We also validated this with a conditional mouse model where we specifically knocked out N-cadherin in smooth muscle. Compared to our control mice treated with the Aspergillus, sensitized challenge with Aspergillus, so these are airway hyperresponsive, we noticed that our N-cadherin knockdown mice had really no airway hyperresponsiveness. And what's interesting is this therapeutic benefit of N-cadherin knockdown is independent of airway remodeling, independent of allergic inflammation. So from all of these data, together with our previous cellular data and slice data, we have localized this response to reducing forces in the airway smooth muscle. Finally, we focused directly in the context of human asthma. We compared ASM cells from asthmatic donors. Actually, these are severe asthmatic donors. They died of severe asthma. And when we compared to normal, we observed more N-cadherin expression in asthma than in the non-asthmatic group. And we also confirmed that this N-cadherin is localized to the airway smooth muscle in the asthmatic lung sections. And finally, we asked the question, why? What is it about asthma that's enhancing N-cadherin? So we took normal airway smooth muscle cells and we treated them with many different known asthma cytokines. And we observed that in the case of TGF-beta treatment, there was a substantial enhancement of N-cadherin expression. So it's only in that case. So this implicates a TGF-beta related mechanism in the asthmatic lungs that invoke this N-cadherin pathway in response. So we've learned in this part, we've learned that N-cadherin is essential for airway smooth muscle contraction, can be targeted simultaneously with airway inflammation, and is able to overcome beta agonism's functional tolerance. Thus, we believe that this antagonism is a robust strategy to inhibit effect in cytoskeleton, fluidize the airway smooth muscle, and ultimately to reduce airway hyperresponsiveness. And more generally, I didn't have time to go through many other detailed studies, but what we have come to learn that this idea of airway smooth muscle fluidization to stretch is a unifying concept that has important therapeutic implications. So that's everything I want to say to you today about the airway smooth muscle, asthma, and contraction. I now want to turn attention to stiffness, another important functional outcome in the lung. Why should we care? Well, through many studies, important contributions by leaders, including Dr. Crapo and many others in the audience and in our community, we've now come to learn that lung stiffening or softening can either be a cause or consequence of virtually every lung disease. And beyond just hindering gas exchange or mechanics on the airway, it also can directly affect important cellular and extracellular matrix function, growth, secretion, division, and even cell death. And it is therefore now a primary therapeutic target across different lung diseases. So, how do we currently measure lung mechanics in drug discovery and in our mechanistic understanding? These are the two prominent methods. One can use an atomic force microscope. So you have a cantilever beam where you touch, or you can use a tissue stretcher. You can cut a piece of lung, hold it between two loading posts, and you can stretch it with a known force, and you can measure deformation and thereby compute a stiffness. The problem is that these methods are slow. I mean, for a little piece. They're complicated. I mean, the adhesion can really affect your outcome. And talk about rigor. Well, here is a situation where it's very difficult to keep staying rigorous between run to run. And these measurements are low throughput. They run one piece of tissue, they run one piece of, one little segment at a time. Importantly, neither of these methods is suitable to studying lung slices. So to overcome this limitation, we developed a methodology called the Mechanobiology. The lung slice, shown here in purple, is adhered to a custom substrate. The substrates that we manufacture are soft, and by soft, I'm referring to kilopascal stiffness. So that's like stiffness of shaving foam, toothpaste. It's very soft substrates. However, unlike shaving foam and toothpaste, these substrates are elastic. That is to say that you take that substrate and you deform it. It deforms with me. But when I remove my deformation, it springs back to where I started. It remembers and it springs right back. So these substrates are elastic. And they're also transparent. So you can then look through these substrates in a microscope to get images. So we custom manufacture these substrates. And we also, at the very top of the substrate, we also embed little fluorescent beads. We then take our lung slice and just stick it to the substrate. And then we take this composite and we press it against these indenters. When it presses against the indenter and gets pulled here, gets pressed, that slice sees a nice uniform stretch. We can run this experiment in a single well format, or we can run this experiment in a multi-well format. And during the experiment, we keep looking at positions of those fluorescent beads that are at the very surface. So in a typical experiment, you can look at that position before stretching and after stretching the slice. And from the relative displacement pre and post stretching, we can compute a map of strain. So how did we strain that material? And then in our paper, we've solved the elasticity equations that with knowledge of that strain map, with additional knowledge of thickness of the lung slice, we can directly calculate stiffness of the lung slice. We first validated this approach with a known material. We chose agarose. We took agarose and we made it at different percentages, 1.5%, 2%, 2.5%. And using the Mechano well, we measured stiffness. So that's shown here with the red circles. I've superimposed on this measurements previously published using either an atomic force microscope, that is, I believe, the triangle, using a nanoindenter, which is this guy, the green one. And this line is using a rheometer. The message here is we see strong concordance between many of these other technologies with the Mechano well. So this predicts known stiffness, and it does it correctly. We then turn to the interesting setting of the human lung slice. We took slices from either a healthy lung and we compared, or a COPD lung, and we compared them. These representative images between healthy and COPD, you can already appreciate the differences in the airway structure. That's apparent here. We then took many slices from here, measured stiffness, and I'm showing you individual slice 1, 2, 3, 4, and corresponding stiffness value. And I'm showing you here individual stiffnesses from many COPD slices. And from these, we computed a PDF, a distribution function. And from here, we then compared side by side. So in blue here is the PDF function for normal, and this is for the COPD slice. What you will observe is two separate distributions. So that's normal, that's the COPD slice. And there is little overlap between these distributions. Also, if you looked at the medians, you will immediately appreciate that the COPD slice is approximately half as stiff as that normal slice on average. But this, what I'm showing you here, is global stiffness. So this is average across the whole slice. What about local single septal wall stiffness? So to do this, we constructed personalized network models. So what we did is we went back to our many PCLS images, and then we developed an algorithm where we could compute alveolar areas. From many of these slices, we then had a distribution for healthy in blue, and then a COPD shown here in red. We then used these distributions and we constructed spring network models. So the area distribution here mimics the real PCLS. This is for the healthy and this is for the COPD. We then would vary the stiffness of individual springs such that the average stiffness of that network matches the real PCLS. So after all of this, we were able to obtain local septal wall stiffnesses. And here is the outcome. In blue here is the stiffness distribution for the healthy. In red here is the stiffness distribution for the COPD. And remember, this is local stiffness. I want to point out two salient outcomes. First, look at this. Look at these numbers. The local septal wall stiffness in healthy is in the order of 30 kilopascal. This is about 20 times bigger than average global stiffness. So the local wall can be 20 times as stiff as global tissue, which I guess makes logical sense. The other point I wanted to show you is you look at these numbers, again, 30 down to about, 50 down to about 25. So it's about the local stiffness in the COPD slices is approximately half to that of health. So how do we interpret all this? Remodeling in COPD reduces local septal wall stiffness, and that's the measurement I just showed you. Now, when we breathe, mechanical forces keep acting on our lungs and on these local walls. So in COPD, you have softer walls. So when you feel the same mechanical forces of breathing, these softer walls will rupture, leading to airspace enlargement. So then you have softer walls in COPD, fewer walls in COPD. Ultimately, you get reduction of global stiffness of the lung. This is how we interpret and how we've used these local measurements in tissue slices to try and understand global lung changes. My last slide on this topic. We were also interested in understanding what are the molecular pathways that drives this remodeling effect. So we did a protein profiling array where we screened 35 proteins that are known to cause ECM degradation. We saw a number of changes, and I'm showing you here the top hit list. So what did I take away from it? Well, to be very frank with you, going into this screening, I didn't know many of these proteins, but I simple-mindedly expected perhaps 1 or 2 or 3 will stand out. But it turns out that's not the case. Several proteins are potentially responsible for this rupture in these COPD slices. But this is interesting and exciting because many of these can now potentially be targeted for COPD therapy. So that's everything I want to tell you about COPD today. I now want to switch gears to IPF. We implemented our methodology of the Mechanobiology in human IPF tissue slices. First, let's look at these representative images. This is healthy. This is IPF. You can see already there is more collagen deposition in the IPF slices compared to healthy. This is also as expected and as is known. And we took many different healthy slices. And here I'm showing you raw data values of stiffness, many different IPF slices, raw data values of stiffness. What we learned from these data is on average, these IPF slices are approximately 2.5-fold stiffer than healthy tissue slices. But this is global. We were also interested in local effects. So we then did local stiffness mapping. And I'm showing you here representative maps of stiffness healthy on top, IPF at the bottom. And to note these very dark colors, the dark black regions are high stiffness values, and you can see big black regions here, as compared to more yellow regions that are the low stiffness value. So this is now in the order of between 5 and 10 kilopascal. Here, for example, is here, for example, is 1 to 2.5 kilopascal. So you can immediately appreciate local stiffnesses in IPF tissues much larger than healthy. We did this again, many slices and so on. And I'm showing you now the PDF functions. What's important to appreciate is compared to the healthy, which is here in blue, you see these broad distributions of local stiffness in the IPF slices. In fact, this variance is approximately 30-fold greater than the variance in healthy. So you could see this very heterogeneous distribution with many local hotspots of stiffening in IPF, 30-fold greater as compared to normal. Okay, so far I've shown you methodologies where it's been method-centric in the IPF circumstance. I've shown you we can measure global stiffness, we can measure local stiffness. Let's now look at its implications. And we first have been looking at bringing these approaches to drug discovery. We started, as just proof of principle, we started with the lab reagent, bacterial collagenase. We took our IPF slices, we measured them pre and post-treatment with this bacterial collagenase. On a slice-by-slice basis, as expected, we saw reduction in the global stiffness, and we also saw reduction in the local stiffness. For example, look at this region here. This is in between about 2.5 to 5 kilopascal, but you can see after digestion that reduces to 0.5 to 1 kilopascal. So this validates our approach as being able to longitudinally measure changes and being able to capture biological effects. We next turned to a clinically relevant compound, nintedanib. We took lung slices, we divided them into 2 groups, vehicle-treated or nintedanib-treated, for 4 days, and we measured stiffnesses after treatments. Compared to vehicle, we noticed that nintedanib-treated slices were approximately 35% softer on average, and this was statistically significant. So our approach was able to characterize, again, I come back to the concept of functional outcomes. We are able to characterize pulmonary function in a dish using human tissue slices. We also were interested in local changes, and I'm showing here representative maps from vehicle-treated slices after 4 days versus nintedanib-treated slices after 4 days of treatment. And it's clear as well that there are local changes. For example, you can see this, which is in the order of 10 to 15 kilopascal, and many of these big hot spots, versus here you can see mostly in the yellows and light reds, i.e. much softer local spots. And when you look at distributions, we observed that the nintedanib-treated IPF slices showed much narrower distribution. So this is what we have expected now for our IPF slices, large variance. But with nintedanib treatment, you can see that that distribution substantially narrows. There's now an 8-fold lower variance with treatment. Interestingly, this is not as small as healthy. So the healthy would be something like this. So we are getting there, but not all the way there. So while this drug is partially effective, it is not your magic bullet. We need better. So far, all these data have established that our approaches are sensitive to global changes, but also local changes. But everything has been focused on stiffness as the outcome. We've now been interested in understanding what underlies this stiffening and or softening changes. So to do this, we are working on combining these stiffness maps with local changes of biology. Here I'm going to show you now ongoing effort. These are preliminary data. This is work together with Dr. Sangeeta Chowdhury's lab at Children's Hospital. What we have done is, after these stiffness measurements, we can slice the slice into little 5-micron thick sections, and we can measure spatial transcriptomic changes. And I'm showing you first changes across all of these ECM proteins. So these are many different known collagen-associated ECM proteins. And I'm showing you here average expression of these proteins across multiple healthy versus different IPF slices of slices. The red are regions of high expression. And you can immediately see that these IPF slices have much more reds, i.e. greater expression of these ECM proteins. Also, they spatially cluster. You can see these reds are all nicely spatially clustered in different regions. We then turn to known fibrotic proteins. And here is a laundry list. And here is the average expression of those proteins. And again, as expected, you see much more of these fibrotic proteins in the IPF as compared to the healthy, and these are also spatially clustering. Another thing that the spatial transcriptomic analysis can reveal is the cellular origins. So what is different between normal versus our IPF tissue slices? I'm showing you here on the y-axis the many different cells that we measured, and on the x-axis is the fold changes in their expression. Let's first look at this region here. This region corresponds to cell types with high abundance in the fibrotic lung slice. And this includes myofibroblasts, fibroblasts, macrophages. So these are retained in our setting and in our experiments. And I'm showing you here cell types with low abundance in the fibrotic PCLS. And these include dendritic cells, alveolar type 1, type 2 cells. These are lost, and this is also well known. But the beautiful, well, the goal for doing this in our context is the slices for which you've seen those stiffness measurements, they also retain known biology. And our ultimate goal is, you've seen these maps of omics, we want to overlay with our stiffness maps. And then we can get a comprehensive understanding, a physical picture where we not only have, well, stiffening, softening, but what's underneath those hot spots? What is changing? What are the cells that are present? What are the transcriptomic changes, proteomic changes in those local regions? This is work in progress, and this is driving many of our exciting studies in the lab. And I will also conclude by saying that we aren't limited to doing these measurements single well, single dishes. We can do them in multi-well, multi-dish formats. We can do them with frozen thawed slices. So these aren't just limited to fresh slices that one can obtain. So we are trying to therefore develop assays that are suitable for, that have the throughput and the outcome suitable for drug discovery. So with that, I will conclude. I wanted to leave enough time for questions. I would like to acknowledge my lab members, a fantastic group of colleagues, a fantastic group of colleagues and collaborators, funding sources, and especially the ATS for honoring me with the Crapo Award. I am just very pleased and energized to keep going. And thank you, all of you, for your attention.
Thank you, Rama, for this very nice, talk. I would like to ask the audience to type your questions on the chat so that we can ask questions to Rama. Let me start, with the one question. Why stiffness rather than simply measure collagen or myofibroblast deposition using histology?
That is a really good question. And in fact, this is something that we've tried very hard to clarify with some of our studies. So when you measure changes of ECM proteins, you have to start with one that you're interested in, the one that you know, you do that measurement. But you also know that just that protein measurement can by itself not inform you of function. The protein, for example, collagen deposition. So a standard for collagen, a standard outcome that people measure is collagen deposition. You can do this with, many standard histological assays. Having more of it does not inform you anything about stiffness because collagen can also cross-link. So similar expression, cross-link collagens is also stiffer. That information is immediately lost if you just do collagen deposition. And I'm sure many in the audience here do these measurements somewhat routinely, and they appreciate the heterogeneity, region to region, slice to slice. How do you put it together? How do you ultimately quantify this to report, rigorously quantify this, to report an outcome? Very hard to do. And also, it is restrictive because it may be your compound, your drugs are not reducing some aspect. For example, alpha smooth muscle actin. Maybe they're not directly reducing myofibroblast clustering, but they're reducing another aspect. So if you just focused on one outcome measurement, you might be missing many others that are also relevant. So my answer to you, David, is in an ideal scenario, you measure not only the biological changes of importance, but importantly, you measure stiffness to understand, do they ultimately translate to something that we can then appreciate in the patient? Something that softens your lung will likely have physiological implications in a subsequent clinical trial. But something that can just reduce a collagen deposition pattern in some circumstances, but not others, may be hard to interpret.
And can you tell me a little bit about nintedanib? I'm very curious because that is a unspecific kinase inhibitor. So how do you feel like that they work on the decreased stiffness in those tissues? What is the proposed mechanism here?
If you waited 3 months, we have a whole manuscript that we're working on to elucidate that. So that's my short answer to you, because not only in order to get to that, to answer that question, these data that I showed you, we picked one dose. We're doing dose response curves. For each of the outcome, we have matched cytokine studies. We've also done histological sections. We're getting proteomic maps to what is changing under these different circumstances. And then we've also done single cell measurements to see what is affected by treatment versus no treatment. So in other words, I can't immediately, because the honest to God truth is I don't know the answer, but it's still work in progress. But we've already found several mechanotransduction candidates to explain how does nintedanib cause softening.
Anyone else have questions?
Well, as people are coming to, may I just make one other comment? I just remembered. Important reason for why stiffness measurements is potentially simpler, integratable, and faster for drug discovery is our measurements are longitudinal and they're non-destructive. So in other words, you can take a slice, you can measure stiffness, do whatever you want to do, treatments and drugs and so on, repeat it, same region, find and remeasure the stiffness. It's not endpoint. And you can do this and you can easily integrate this with histology for the same slice. So bottom line is you can longitudinally look at drug treatment circumstances.
In homage of Dr. Crapo, that is the name give this award, how you envision that measure stiffness can eventually translate in the clinic? I mean, how potentially doctors can measure stiffness in the lung?
Well, they don't have to necessarily measure stiffness in the lung, but we know that stiffness is very closely correlated to any lung mechanical measurement, FEV1, so on and so forth. So this is as close as possible currently to a real in vivo correlate of respiratory function. So something that softens is expected to enhance lung mechanical outcomes.
We got a questions from the audience. Dr. Heiss is asking that, number one, she said this was a great talk. And can you measure viscoelasticity with the mechanowells?
Good question.
Do you think that will change with the treatments?
Very good question. And that's also work in progress. Another project is precisely that, to add dynamics to our measurement, to look at potentially different frequencies and get your question is spot on to what we are thinking. And that's especially relevant. And I've shown you here an example. We do lung mechanics. This was all implemented with lung slices. This can be used with really any tissue slice, organoid models, 3D materials. And this is something that we've discussed many times and which we want to do. So the short answer is yes. The more difficult answer is, but has not yet been accomplished. So unless I demonstrate it to you, I can't tell you it's feasible, but we are working on methods to get there.
We got a second questions from Dr. Theodorescu. And how do these in vitro measure how these in vitro measures relate to in vivo measurements such as elastance and compliance? I think it's very close, but can you explain a little bit about this?
Right. I mean, that's kind of what I was trying to say is it is closely correlated in the sense that stiffness will determine how the lung can stretch and unstretch with in measurements of elastance and compliance. We have several specific examples that we have stated in that manuscript, but I can't quite remember them. But for the sake of this kind of presentation, I would say it is a very close correlate, and it's very informative to what you can expect with elastance and compliance measurements. And I will say one other thing I noticed in the audience is Dr. Rachel Knipe. So we are working on a project together where where Dr. Knipe's group has measured elastance and compliance in mouse models exposed to different conditions and treatments. And we would like to overlay that with our stiffness measurements directly, so we can provide to you a more specific answer than my kind of, bigger picture theme. But that's also another important project that we're working on. It's a way also to not just match the data, but also to understand the outcomes from mouse models.
The one question I have here is, for the point of view of drug development, in many cases, the drug focus on immune cells, epithelium, maybe some muscle cells, but always we forgot about the myofibroblasts. Do you think those could be potential good targets based on?
Oh, no doubt. In fact, there are several companies and efforts, and I was at an IPF conference recently. This is a major theme of what people can measure. And in fact, when I did those omic maps, you can see clearly myofibroblast clustering, approximately similar sizes to our stiffness hotspots. And there is a lot of biomechanical literature. I've also done some of these studies in cultured cells where it makes perfect sense that those hotspots may be defined. Those cells might be defining either in or around those hotspots. So that certainly is an important point to consider. And I will make yet another point related issue there is another largely ignored but important cell to study is the endothelial cell. And how do those cells contribute? So in these lung slices, we have preserved vessels and preserved endothelial cells. Those contributions can also potentially be studied.
Aleksandra, do you want to ask any question? James?
Yes, I'm waiting for my turn. Yes, Dr. Krishnan, I have a question about barrier integrity. So does N-cadherin targeting affect E-cadherin and barrier integrity in bronchial epithelium?
Very good question. I should have clarified this. They're homologous. N-cadherin is present really just in the airway smooth muscle. E-cadherin is predominant in epithelial cells. So my whole talk was not talking about E-cadherin. You don't want to antagonize E-cadherin because that's going to cause barrier disruption and so on and so forth. So these are specifically targeting N-cadherin, not the E-cadherin component. But that's really an important point I should have clarified.
There's a question from the audience, which is a very good one, from Dr. Kristen Warrington, is how did you define distinct cell population within your samples? For example, how did you decide what makes a myofibroblast versus
good point.
cell type? This is one big discussion in NIH right now where we try to name different cell types in the lung.
It's an excellent question, and I can tell you this is still emerging collaborative work with Dr. Chowdhury. This is sort of her wheelhouse, but I can tell you that referred to the single cell atlas. That's widely available. That's how we determined and apportioned ourselves populations.
And just for clarify that the population is not completely well defined, it's still NIH have this consortium of the scientists across the globe try to define the molecular markers of different cell types, including the novel cell types that come from the single cell RNA sequence and the spatial transcriptomics.
Good point. And since we are on this topic of cell populations, one of the important things for us as well in this whole effort was to characterize these cells, and you've seen some omics maps. These were all done in cryopreserved thawed slices. That's really important because I want to make a case here that, historically with lung slice-based studies, one would wait for a lung to arrive. A lung arrives, you can get hundreds of slices. In fact, there's technology now, you can get thousands of slices. But again, historically, the measurement was limited to, it started with 3 days, extended to 2 weeks, and now I have seen papers that can push it to a month. So you can do studies. You waited, you've gotten a precious lung, you slice, 5, 6 people come together, 8 people, 10 people, but, if you want to make thousands of slices, gotten all these slices, but then you're limited, your time window is limited to at most 30 days. This is a major limitation, right? So one of the other things that my lab and others have made a case for is cryopreservation. You obtain these hundreds of slices. We have published methods, very straightforward methods, where you can immediately cryopreserve them. And then on demand, you can thaw them out, and it still retains many of the important things we want to measure. For example, I have looked at fibrotic slices, recent paper together with Dr. Holger Bersing's group at IIBS. We compared fresh versus frozen thawed. We looked at it over 14 days, and we looked at production of cytokines. We looked at stiffness. We looked at induction of fibrosis. Many of these things were preserved and similar to fresh. And all this omic mapping that we did were all in these cryopreserved thawed slices. And we saw, again, known biology preserved.
So we only have like 2 minutes left over. So I would like to thank you everyone for attending in behalf of the respiratory structured function and the web committee that is being basically hosting this event. Do you have a final remarks, Rama?
Oh, I'm glad everybody attended, and I hope you had an informative and useful time.
Thank you very much, Rama. We congratulate you again with the award that was featured today in our live webinar. Thank you very much for joining us, and thank you, Dr. James Crapo, for being with us today. Thank you, Dr. Crapo, and have a great day, everyone.
Thank you.
Bye-bye.